Thanks for staying. It's always difficult to be the last speaker after the lunch has all gone. And also, I feel a bit like one of these xenocells. Really, I'm a human genomicist, so slightly out of water. What I'd like to do is take you through some of the things people have used the solid system for, and also to then at the end take you through the technologies of both solid and the ion torrent. So we really live in a truly amazing time. To me, actually sitting here and listening to you all talk about reprogramming cells and then the inevitability that that will eventually be used in treatment really just makes me think we really are in the century of biology. Since the Human Genome Project was finished, which, by the way, if anyone ever tells you that this project was a waste of money, there's a recent report that showed it cost $2 billion and generated $179 billion so far in economic returns. Don't let them tell you that. But it's 10 years since we finished. And what's that actually done? That actually means that now a lot of these procedures are actually very routine. A couple of things I always like to pick out. This is a very nice chart from Nature. One is that scientists have a sense of humor. One of the genomes they sequenced early on was the platypus genome. The world is changing incredibly. Consumers are becoming very, very DNA aware. This is the biobank in the UK. 500,000 people have signed up for the biobank, despite the fact they are told when they sign the release forms that they won't benefit them at all. This is for future generations. 500,000 people is over 2% of the population of the UK. People really are into DNA. And we've done even some amazing things now where by sequencing individual people, we've been actually able to determine the genetic reasons for complex genetic defects, like, for instance, mild mental retardation. So the challenge is that none of us are biologists anymore. When I started doing my stuff, we were all working on, one gene. It took you a week to sequence a whole gene. But now we actually need to integrate everything together. You can actually do everything on the solid system. You can do your sequencing, you can do your copy number variation, you can do your RNA, you can do your epigenetics, you can do your RIP. You've heard about the chip. You can do literally everything on the solid system. So I'm going to talk, really, I split it into 4 areas. I'm not going to talk about de novo sequencing. I'm not sure if that's a big pull for this audience, but in actual fact, in the cancer world, people, even with a reference, are interested in de novo. But resequencing, transcriptomics, and an epigenetics. So resequencing is obviously when we actually have a sequence out there, and we just do the sequencing and assemble it. And I'm sure you're all aware, many individual genomes have been done. This is actually our chairman at the bottom. Probably up till recently, the most famous genome was Archbishop Tutu. But actually Ozzy Osbourne's been done, which it turns out is much more widely known worldwide than Archbishop Tutu, which is somewhat sad. I think you can all realize what you would do when you sequence an individual genome. This is a great way to actually get all the information about your cell line. But I think one thing that might be interesting is to know what happened in the cancer field. We're all used to looking at cells and looking at the SNPs, or maybe measuring small indels or microsatellites and saying, yes, this is the same cell. When the cancer people actually started sequencing whole genomes using next genome technologies, the next generation sequencing technologies, the first thing they realized was there was a much, much higher level of rearrangement than they expected. And this is actually shown in the diagram on your right. This is actually the rearrangement. It's copy number variation. There's actually an insertion and there's an inversion, all in this one small area of chromosome 1, none of which showed up on the Sky map. And this they've seen again and again, that when you can look at the small scale, look right at the individual base level, much more rearrangement. So I think that may be something that people will want to look at when you're actually telling if your cell lines have changed, is to actually also go in, do whole cell studies, whole genome studies, to actually look at whether there's actually been rearrangement. This is an example of 15 different cancer cell lines, and you probably can't see too well, but what you see very clearly is there's no distinctive rearrangement that stands out. This is the rearrangement for this cancer cell line. What you see is lots and lots of different rearrangements, some cells having much more rearrangement than others. Very, very important, and you literally are down there at being able to measure this literally at the base level. And you do this very simply by just doing what we call mate pair mapping, comparing 2 tags where you expect them to map with what you observe, and then you can actually make inferences. It's actually incredibly simple. It sounds like it should be complicated, but in fact all we do is list 3 letters for the status of your mate pair library, and using that you can deconvolute these extremely complicated rearrangements. I slipped this one in just because I wanted to mention this. This is basically exome resequencing, and this has been the big thing that's happening at the moment in the human disease world and even in the cancer world. Because if you sequence an exome, you're doing 3 to 5% of the whole genome. But really, for many people, they say it makes the analysis easier. Because if I sequence the whole genome, I'm going to get an awful lot of differences when I compare to the reference that I just don't know what they are. They're in non-coding regions. They're in regions I know nothing about. What can I tell? If I do the exome, I can at least make predictions on whether there's protein changes. And this has become very routine. In fact, it's so routine, there was actually a paper out on Thursday, so I couldn't actually put it in this, where in fact exome sequencing was used to change the treatment of 2 children, and those children now have a much better quality of life, literally based on exomes, on genome sequencing and pulling out what genes were present. But what you do is you sequence the exome, you look for the SNPs that are present. This obviously be a very good way of comparing different cell lines, and then you make conclusions. But what's amazing is we literally live in a time when, by using a single individual, we can actually work out the genomic disease that they have. So again, I think the power of exome sequencing, the power of complete genome sequencing, is something that really hasn't yet been taken up, as far as I can see from the literature, by the stem cell community. So really, I think that the first target, and we've heard this already from Ron, he talked about the ChIP-seq experiment, is really the gene expression. Why would I do gene expression on a next-gen sequencer? I usually do this bit of the talk when there's a salesperson in the back of the room, and usually by the time I make my first 2 points, they've gone white and you can see them shaking. Next-gen sequencing is still slightly more expensive than doing an expression array, especially when you consider you've already got the infrastructure set up in your lab and there's changeover costs. In fact, I was talking very recently with a core lab who actually said using the barcodes, they've actually now got their next-gen sequencing gene expression experiments the same cost as actually doing an array. The analysis is much more routine with a gene expression array, and I think this is something we have to admit, and that's partly because it's been going along for 10 years. We're really at the stage, the gene expression arrays were about 2 years into their existence. There are a lot of techniques out there. People are starting to standardize on what are the ways to do the measurements, but it's not completely standardized, it's not cookbook. So by now you're thinking, why on earth are you saying this? Why would I want to do gene expression on a next-gen sequencer rather than an array? And usually by now the salesperson is a quivering wreck in the corner. The reason is, if you do it on an array, you can only find the sequences that are on that array. And usually those arrays come from commercial vendors, so you're relying on someone else's bioinformatician to actually put on that array what you need to look at. An example is the long non-coding RNAs. They've only just started to appear on commercial arrays, and yet they've been in the literature for over a year. If you do next-generation sequencing, as long as when you do the library step you capture the RNAs, you will see them when you do the sequencing. It's also digital. So if you need to do more sensitivity, you can do more sequencing and add the results together. There are some statistical methods where you can improve calling by using many, many arrays, but you can't actually add the results together to increase the sensitivity. I think the real kicker is when I speak to people who have shifted over to doing next-gen sequencing, using that to do their RNA expression analyses, they're telling me that they see 30 to 50% of the peaks they see were not on the arrays that they would have used. So essentially, if you don't use next-gen sequencing now, you know you're going to actually miss a lot of information. So I want to show you what some of those things that have been done. The first is just that little point to remind you this is all done on the same platform. You do your whole genome sequencing, you do your expression sequencing on the same platform, and I'll actually point out how that's the case when I get to the machine, that you can actually do them in the same run. This is actually a tumor from an oral tumor. We actually did whole genome sequencing and we did whole transcriptome sequencing, and we're able to actually correlate the gene expression levels to the copy number variation. This is actually quite interesting because in terms of the people in this room, if we were actually to look at the number of bases we differ by, not the number of events but the number of bases, the biggest difference between me and any one person in this room well, between me and the men in this room, obviously there's one big difference between me and the ladies, is, and you can laugh, is that it'll actually be copy number variation, because each of the copy number regions can be in the hundreds to thousands of bases long. But this is next-gen sequencing, so we can go right into the individual base level. So what you're looking at here is the forward and the reverse in the normal and the tumor. And I don't know if I have a cursor. No. If you look here, you'll see this little tiny peak here and this little gap here. Ooh, can't do that very easily. If you didn't have strand-specific sequencing, you wouldn't be able to tell those two apart. And that's really the advantage of strand-specific sequencing. And in this particular case, we're actually able to spot an overexpression in a region of copy number variation that's actually an oncogene. We've already heard about the microRNAs from Ron. This is my favorite sort of paper, where in fact you can look at one diagram and you really don't have to read the rest of the paper, even though it turned out to be very interesting. This is actually differentiating two types of cancer. One is favorable neuroblastoma and one is unfavorable neuroblastoma. Before you run out being hopeful, you don't want favorable neuroblastoma. It's still a nasty disease. But unfavorable neuroblastoma does not respond to existing treatments. So if you look here, what they've done is they've actually taken the microRNAs, they've sequenced them on the solid, and you really don't need to have any degree in statistics or any R packages to see that there's a difference between these 5 cell lines and these five samples. The unfavorable and the favorable very clearly discriminated. Now, to be honest, if I was then going to put this into an assay, I wouldn't do the assay on the solid system. I would take these specific microRNAs and turn them into a TACMAN low-density array assay, would be the way I would do it. They're also able to actually, because it's base-to-base sequence, look at things like RNA editing events. Even with the existing huge number of microRNAs in the database, they still found another 13 novel microRNAs, 3 of which they followed up because they followed this pattern and they were all confirmed. So very, very exciting, and it really shows how you can differentiate cell lines using next-gen sequencing. This is one that just recently came out from Stanford. PARS, for any young researchers in the audience, before you start any experiment, make sure you have a really good acronym. That's the way to be successful. And what you do here is what we've been doing before, and this is actually the biggest thing that next-gen sequencing does. What does next-gen sequencing do? It delivers you lots and lots and lots of data. So that's it. So what you can do is take techniques that you used before and put them on steroids. So previously we've taken single mRNAs, used restriction enzymes to actually work out RNA structure. This is essentially doing the same thing across the entire transcriptome. And when you look at everything together, surprise, surprise, you find the coding sequences are more likely to be double-stranded, and the start and the stop points are essentially open, which is single-stranded, which is exactly what you would expect. This is looking at viral transcriptome and host transcriptome at the same time. They're very important for the medical field. Instead of looking at these separately, this is within the same culture solution. We're looking at these, and what we see here is the viral transcriptome. These are the time points in 2 different experiments. For the cell line, despite the fact this is cell line, so it should be identical, there's actually a 25% difference between these 2 cell lines, and I don't think I need to tell this audience that human cell lines are very annoying in that way. They show a lot of variety. This, to me, is one of the most amazing things I've ever seen. There's 2 things I think I'll always remember in science. And one is when I saw the cover of Science where they actually had a picture of an atom. And the other is when Kai Lau came down and said, oh, we can do transcriptomics of a single cell. So this is literally going in and looking at the transcriptome of a single cell. The bottom paper here is actually where we prove the point, and we actually show that it's much, much more efficient than arrays. If you're interested in doing it, this is the protocol, very detailed. I strongly suggest you read it. It's got a lot of hints on how to work with the single cells. And then this paper here, they looked at 33 different stages between inner cell mass and embryonic stem cell in the mouse, and actually able to produce a pluripotency map. So in the interest of time, I'll skip straight to that. So one of the ways they were interested here is they wanted to look at not what genes expressed, but what actual transcripts are expressed. How do we actually look at the isoforms of the transcripts? How do we do this with these short sequences? We're sequencing here 50 bases. We're not sequencing kilobases. We don't get the whole transcript in one go. So what you do is you actually work out which are the exon junctions that make the transcripts unique. And then you look for the counts of the crossing of those exon junctions. And as you can see here, in the embryonic cell mass and the inner cell mass, this gene is expressed, but there's actually basically a flip, a transition between the two isoforms expressed, whereas in the epiblast, there's essentially no expression. Very, very powerful methodology. Methylation analysis is something that you can do by sulfite conversion, capture it and enrich it, or enzymatic. This is an example of capturing with methyl miner, which is a kit that pulls down methylated regions. This is an enzymatic method. You basically split your sample into two, and you use methylation sensitive and methylation requiring enzymes, and you get a pattern like this. You actually look across the methylation across the whole genome. Very, very powerful. And in fact, this group actually found some patterns that they actually are publishing a second paper on, which they link to specific types of cancer. ChIP-seq you've already heard about. If you have any questions, please just ask. So, very quickly, the new genetic analyzer, the new solid. I forgot my prop. The new solid, we've just released. What we essentially did was we went to our customers like Ron and we said, what are the things that are annoying you? And we remade the machine. The key thing we've done is paper lane sequencing. Many, many people come back and say, you know, I don't have 200 gigabases or 100 gigabases of sample this week, but I want to run the machine because I need the results from this experiment. So what we've done is we've taken the slide and split it into 6, and you can load each lane individually and pay just for the chemistry to run that individual lane, which means you can actually put any application you want on the lane. So literally, you could do a transcriptome experiment, a whole genome sequencing experiment, a targeted resequencing experiment, small RNA, all on the same chip, and just pay for those reagents that you need, which is one of the big problems with next-gen sequencers. They really were designed when they first came in for the very, very large labs. And we've been amazed how they've moved into the single investigator labs. The other thing we've been doing is just automating. The front end is becoming the challenge because the sequencing machine is actually pretty simple to use nowadays, but you've got so much throughput, 100 gigabases of throughput, that's many, many microRNA experiments. So automating the sample preparation, the library making. We've also automated the informatics. For many labs, this is the scariest thing there is informatics. So we've automated that with LifeScope, which basically has a nice web-friendly interface. It looks like you're going through the web to your server. It takes you through these workflows that tell you you need to add a reference, you need to add a sample, you need to set up the analysis. Takes you through. You don't need to use Unix. I'm always amazed. I go to labs where people are literally doing single-cell work, which I think is amazing. I never myself work with single cells other than bacterial. But as soon as you ask them to type one Unix command, they panic. And Unix is just English without vowels. Oh, you all use it then. We make the results viewable with graphs so that you can visualize them easily. That we're all visual creatures. We also make sure you can export them to the most commonly used bioinformatics tool in the world. It's still Excel. Visualizing in third-party viewers. This is the IGV Broad viewer. And we'll also sell it pre-configured on a workstation. This workstation fits beside your desk. You just plug it into your local network and you're ready to go. And that workstation is more powerful than the cluster we sold 6 months ago. So, very last few slides, Iron Torrent. It's our latest baby. And what it does is it uses these chips. It's the cheapest sequencer you've ever bought. It's the most expensive pH meter, because that's what these chips are doing. They're measuring pH. It's benchtop. Very fast, 2-hour run times. Very simple. As I say, it's just a pH meter, no fluorescent molecules. You're just using polymerase natural nucleotides. You can actually do an entire run from sample prep to analysis in a day if you prefer, if you like a 10-hour day. 2 days from DNA to run if you like your lab technicians. The chip is the machine. This is a 314 chip. You can buy this today. The 316 is out very shortly, next month, and the 318 will be out August, I believe. 10 megabases of data sequence off this chip, 100 megabases off the 316, 1 gigabase off the 318. All the chips fit in the same machine, no change to your workflow, nothing. All the complicated stuff happens on the chip. The chip is measuring the hydrogen that's released when you incorporate nucleotides. So if you know how a 454 sequencer works, it works essentially the same way, but it doesn't have an optical system. As I say, it's just a pH meter. We flow DNTPs across, and we measure their incorporation with an ionogram. Multiple applications already being done. As we move to those larger chips, I think you're going to see the applications that you want to do being available. Amplicon sequencing, though, if you want to actually look at specific pathways, very, very popular already. RNA sequencing to look at highly expressed genes coming with 318. And the chip sequencing will be being done with the 316 and the 318 chip. So with that, I'll say thank you.